Welcome to our lecture on the neuromuscular junction. Today, we are going to explore the fascinating biological bridge that translates an invisible thought in your brain into a powerful physical movement of your body. This is a fundamental concept in human physiology, and by the end of this video, you will understand exactly how your nerves and muscles communicate. To begin, let us imagine you are a brilliant military commander standing on one side of a raging river with your loyal army on the opposite bank. You cannot cross and they cannot hear you. So you write orders on paper airplanes and throw them across. In your body, your nervous system is the commander and your muscles are the army. The river is a microscopic gap between them, and the paper airplanes are chemical messengers. This entire setup is exactly what we call the neuromuscular junction. Now that we have an intuitive understanding, let us look at the formal biological definition. The neuromuscular junction is a highly specialized synapse between a somatic motor neuron and a skeletal muscle fiber. The very end of the nerve, the synaptic terminal, holds thousands of tiny sacs filled with a neurotransmitter called acetylcholine. Across the gap, the muscle has a specialized receiving area called the motor end plate, packed with receptors. The fluid-filled space between them is the synaptic cleft, where the chemical exchange happens. Before we move on to the clinical side, let us see this structure visually. Here, you are looking at a schematic of the neuromuscular junction. At the top, the yellow bulb is the nerve terminal. Inside, you can see those circular vesicles holding the blue acetylcholine molecules. When an action potential arrives, these vesicles fuse with the edge and dump their contents into the synaptic cleft, that empty space in the middle. The pink area below is the muscle's motor end plate. Notice how it is folded to increase surface area and packed with purple receptors waiting to catch the acetylcholine. With the anatomy clear in our minds, we must ask why this specific junction matters so much in real life. It is absolutely critical because it is the sole mechanism controlling your voluntary muscle movement. Every blink, breath, or step relies on millions of these junctions firing perfectly. If this communication is severed, it leads to total paralysis. We see this in autoimmune diseases like myasthenia gravis, where the body mistakenly destroys its own muscle receptors. Even deadly poisons like black widow venom or Botox work specifically by hijacking this tiny gap, proving how vital it is to our survival. Understanding the biology leads us to how we can measure this electrically. When acetylcholine binds to receptors, it creates a tiny electrical spike. But one spike is never enough. The total electrical change, called the end plate potential, is found using a simple formula. E equals N times Q. Here, E is the total potential in millivolts. The variable N is the number of chemical packets, or vesicles, released, and Q is the quantal size, the tiny voltage from just one packet. If the nerve releases more vesicles, N increases, and the total potential E increases, making contraction much more likely. Now that we have our formula, let us apply it to a real biological scenario. Suppose a nerve terminal releases 80 vesicles of acetylcholine, and each vesicle produces a miniature potential of 0.5 millivolts. We need to find the total electrical change. We start with our formula, E equals N times Q. We substitute 80 for N, representing the physical packets, and 0.5 for Q, representing the voltage per packet. Multiplying 80 by 0.5 gives us 40. This means the total end plate potential generated by the nerve signal is exactly 40 millivolts. This calculated potential brings us to our next crucial step. The end plate potential we just found does not exist in a vacuum. It adds on top of the baseline electrical state of the muscle cell. To find the final state, we use the formula V final equals V resting plus E. V final is where the muscle's electrical state ends up. V resting is the initial negative voltage of the resting muscle, and E is the massive influx of positive charge we just calculated. If E is large enough, it pushes the final voltage past a critical threshold to trigger a contraction.
Let us continue our previous example to see if the muscle actually contracts. We are given a resting potential of -90 millivolts and a critical threshold of -55 millivolts. We take our calculated E of 40 millivolts and add it to the resting potential. -90 plus 40 gives us a final membrane potential of -50 millivolts. We must now compare this to the threshold. Because -50 is more positive than -55, the signal is strong enough. The muscle will successfully contract and generate physical force. So we have successfully triggered a contraction. But what happens next? After a muscle contracts, the acetylcholine must be rapidly cleared from the synaptic cleft. This is essential so the muscle can relax and prepare for the next signal. An enzyme called acetylcholinesterase acts as the cleanup crew, breaking down the acetylcholine into inactive pieces. If this clearance process took too long or failed entirely, the muscle would remain locked in a continuous state of contraction. This would lead to severe muscle spasms or even suffocation if it involved your breathing muscles. To understand just how fast this cleanup happens, we can calculate the clearance time. The formula is T equals N divided by R. This is a classic rate problem, just like calculating how long it takes to drain a bathtub. The variable T represents the total time required in milliseconds. N is the total number of acetylcholine molecules currently floating in the gap. And R is the rate of enzymatic breakdown, measured in molecules destroyed per millisecond. By dividing the total amount by the speed of the enzyme, we find the exact duration of the cleanup. Let us put this formula to work with some biological numbers. Suppose there are 10,000 molecules of acetylcholine currently in the cleft. The enzyme breaks them down at a rate of 4,000 molecules per millisecond. We want to find how long it takes to completely clear the gap. We substitute 10,000 for N and 4,000 for R. Dividing 10,000 by 4,000 gives us 2.5. It takes exactly 2.5 milliseconds to clear the neurotransmitter. This incredibly small fraction of a second highlights just how rapidly biological enzymes operate to keep us alive. We have covered a lot of ground today, so let us review the most important ideas. First, the neuromuscular junction is the vital communication bridge between your nervous and muscular systems. Second, it relies on converting an electrical nerve signal into a chemical messenger, which then triggers a new electrical signal in the muscle. Third, we saw mathematically how the total end plate potential depends on the number of chemical packets released. Finally, rapid clearance by enzymes is absolutely vital for normal movement and survival. You now have a solid foundation in muscle physiology.